So hello everyone, our today's topic is on physiology. So let us discuss the first question. Which of following it is involved in cell adhesion and it is composed of transmembrane glycoprotein? The answer would be cadherins. Well, the cadherins, remember these cadherins, they are number one, transmembrane glycoproteins. So what are cadherins? So these are glycoproteins that play a crucial role in the cell adhesion. So they play a role in cell adhesion. So they are involved in formation of adherens junction, which are cell-to-cell junction that provide mechanical strength and facilitate the communication between the cells. Cadherins, they are involved in cell adhesion and they are composed of which protein? Transmembrane glycoprotein. So imagine you have a bunch of building blocks. Cadherin, they are little connectors that hold the blocks together. So they make sure the blocks, they stay attached to each other in a right way. So similarly, you have a stack of building blocks. So these cadherins will make sure that these blocks stay attached to each other in a right way. Similarly, in your skin, these cadherins would help skin cells to stick tightly to each other. So if your skin does not, so that your skin does not fall apart. So cadherins, they are basically the cellular glue. What are cadherins? They are the cellular glue that ensure that our body cells, they stick together and work as a team. So cadherins, they are like a glue that helps the cells to stick together. They are the proteins that are found on the surface of cells. So they use, remember, they use calcium ions to work properly. So when the cells, they have same type of cadherins, they stick together more. For example, one cell that has, suppose one cell has a cadherin, and it will stick together to other cells that will have a cadherin. So think of cells as people and cadherins as the handshake that makes them friends. So if two people use the same handshake, they will become close friends and hence they will stick together. So if the people use different handshake, means different cadherins, they still can be friends, but they might not stick together as strongly. So these cadherins are just like the handshake that helps the cell becomes friends and stick together. Whereas the other option, microfilament, they are composed of the protein. The microfilaments, they are composed of protein that is actin. So they play a role in cell structure, cell movement and cell division. So the microfilaments, they are involved in process like muscle contraction, cell motility and maintenance of the cell shape. Microtubules, they are other component of cytoskeleton. They are made up of tubulin protein. So these microtubules serve as track for the motor proteins like kinesin and dynein that transport the cellular material. Whereas the intermediate filament, they are composed of various type of proteins like keratins, pimentins, etc. So they will provide mechanical support and maintain the structural integrity of cells and tissues. So which of following cells they are involved in cell adhesion and it is composed of transmembrane glycoprotein? The answer would be cadherins. Hope you understand that. Let us move to the next question. The next question is which of following organelles, cell organelles, it is responsible for synthesis and packaging of lipids? The answer is endoplasmic reticulum. Well, the endoplasmic reticulum, it is network of membrane. The endoplasmic reticulum, specifically the smooth endoplasmic reticulum, it is responsible for synthesis and packaging of lipids and production of phospholipids and steroids. The Golgi apparatus, it is primarily involved in modifying and packaging proteins, while the lysozyme, they contains enzyme for intracellular digestion. So remember, this endoplasmic reticulum, it is just like a factory. So it is a factory where the proteins are made. The ribosomes, these ribosomes are the workers. And remember, mRNA is the instruction. So these products, they can be either used inside the factory or they can be sent out. Just like proteins can stay in the endoplasmic reticulum or be transported to the other part of cells or the body. So which of following it is responsible for synthesis and packaging of lipid? It's the endoplasmic reticulum. So remember, the Golgi apparatus, it is responsible for modifying and packaging the proteins and lipids, whereas the endoplasmic reticulum, it is involved in protein synthesis and lipid synthesis. The lysosomes, they are responsible for the intracellular digestion, and peroxisomes play a role in lipid metabolism. Coming to the next question. So which of following cells junction allow for direct communication and passage of small molecules between the adjacent cells? The answer would be gap junction. So what are gap junction? Gap junction, they are the communication tunnels between the cells. So these are the gap junctions. So they are just like the communication tunnels between the cells. So they are the channels that allow small molecules. So it will allow small molecules like ions or electric signals to pass from one cell to other. So gap junctions, they are like little tunnel that connects the cells, enable them to exchange information and coordinate their activities. So they are just like a cell phones. So gap junctions are like small cell phones that use cells to talk to each other. So they help cells share information, molecules, and even the electrical signals. So inside these gap junction, there are special proteins that connect one cell to another. So these proteins are known as connexin. So they are just like a phone number for these cell phones. These connexin, multiple connexin, they connect together to form connexin. So these are just like walkie-talkies that allow cell to directly talk to each other. So gap junctions helps with the transmission of electric signals between the nerve cells. So they also let the molecules and ions pass between the cells. The cell junction that allow for direct communication and passage of the small molecules between the adjacent cells, they are the gap junctions. Desmosomes, well, they are just like the sport welders. So they are specialized structures that will hold the cell together and provide strong mechanical connections. So the desmosomes use protein filaments to attach one cell to another. The tight junctions, they are like seal that prevents the leak between the cells. So they create a tight barriers between the cell, preventing the substance from leaking between them. Think of tight junctions as Ziploc bags, keeping things from spilling out. Hemidesmosomes, they are like anchors for the cell to their surroundings. So they are like anchors that keep the cells in place and maintain the tissue integrity. So the desmosomes, they act as port welders. Gap junction allows for the communication tunnels between the cells. The tight junction, they act as a seal to prevent leak. And hemidesmosomes, they are like anchor that attach cell to their surroundings. So your answer would be gap junction here. Let us move to the next question. It was asked in one of your AIMS examination. RBC in the blood cells, they repel each other. Well, the repulsion of the red blood cells, it is due to the negative charge. So the negative charge on the carbohydrate moiety of the glycoprotein in the RBC membrane, particularly the glycophorin A, that is heavily modified by these sialic acid residues. So these negative charge group, they will create a electrostatic repulsion between the adjacent RBC. So that will prevent them from sticking together or aggregating. So that is important for maintaining the fluidity and flow of blood in the circulatory system. So you can just remember it by consider each RBC as a tiny donut. So on the surface of donut, there are something glycoproteins with negative charge, like the tiny magnets that push them away from each other. So keeping the donut from sticking together, so that your blood flows smoothly between the RBCs. All are repelling each other, staying separate and not clumping up. So that is important because it will help your blood move easily through the blood vessels. So that is important so that your body gets oxygen and nutrients it needs. So thanks to this tiny magnet charge on your RBC, that prevents them to clumping. So the answer would be C. So because of carbohydrate moiety of glycophorin A in RBC membrane that have a sialic acid residue, that is why the RBC in blood repel each other. So this question was asked in recent AIMS examination. Hope you got the explanation. A polysaccharides coating that cover the outer surface of many bacteria and allow the bacteria to adhere firmly to the structures is glycocalyx. So the option are pili, glycocalyx, fimbriae. The answer would be glycocalyx. So glycocalyx, it is just like a shield for bacteria. So it gives two functions. One is the protection and other is the adhesion. So protection, it will act as a protective coat around the bacteria. So it will so suppose Superman has a super suit. So this one, the glycocalyx, would act as a suit for the that shield the bacteria from the threats in the environment. Second, these glycocalyx also work as a glue, so that will allow the bacteria to stick to surface whenever, whether it's in your body or an object. So this stickiness, it is vital for bacteria to survive. So the answer would be glycocalyx. Coming to the next question, the coagulation time, it is prolonged in. Well, the coagulation time, it is prolonged in hemophilia. So hemophilia, we know it's a genetic order that would affect the blood clotting. So people with hemophilia would have deficiency of certain proteins that are known as clotting factors. And have a tendency to bleed excessively even from the minor injuries. So that leads to increased coagulation time, as the blood will take longer time to form clots. So the coagulation time, it's prolonged in hemophilia. Coming to the next question, the pernicious anemia, it is caused by the deficiency of it is caused by the B12 deficiency. So pernicious anemia, it is caused by deficiency of the intrinsic factor. That is a protein. It is a protein that is formed in the stomach. So this intrinsic factor, it is responsible for absorption of which vitamin? Vitamin B12. So when there is lack of intrinsic factor, it can lead to inability to absorb vitamin B12. So that in turn would cause pernicious anemia. So vitamin B12, we know it's an essential nutrient your body needs for various functions, including making RBCs and maintaining a healthy nervous system. So when you eat food containing B12, it will go into stomach and your intestine. So intrinsic factor would play a crucial role here. So it acts as just a guide, as a tour guide, escorting vitamin B12 through the lining of your small intestine and your blood stream. Think of intrinsic factor as a guide that will lead you through a museum. So now if there is a problem with the intrinsic factor, the B12 cannot get through the lining of your intestine. So the tour guide is missing and not doing its job. So without enough B12, your body will not make enough RBC. So this shortage of red blood cells, that would lead to condition that is known as pernicious anemia. Anemia means that you have a fewer oxygen carrying red blood cells in your body. So that can lead to symptoms like fatigue and weakness. So hope answer is clear. Moving to the next question. The urea and creatinine excretion indicates early renal damage. Urea and creatinine excretion can indicate early renal damage. So increase in level of urea and creatinine in the urine may suggest that kidneys, they are not functioning properly, and there may be a kidney damage. So that can give an early indication of renal problems. The base of heart it is formed by, well, the base of heart it is formed by right atrium and left atrium. So the base of heart it is remember the top part of heart, and it is separated from the rest of thoracic cavity by structures like esophagus, aorta, etc. So think of heart like a house. The base of heart is like the roof of the house. It's the top part. Now the roof is mainly made up of left atrium, which is one of the heart's chamber. Coming to the next question, when a patient pulse is being taken, his arms should be well supported and above his shoulders. So extending and positioning at the same level or lower than that of heart. So this position is so that ensure a accurate measurement of the pulse. So placing the arm in different positions, they can lead to inaccurate reading. So the arm should be at the same level or lower than that of heart. Which of following contributes to the osmolality of the plasma? So among the options, the proteins they contribute to least to the osmolality of the plasma, while the glucose, sodium, urea, they helps to determine the plasma osmolality. So osmolality of plasma, it's measure how many concentrated or diluted your blood is. It depends upon things like what is dissolved in your blood, like salts, sugars, proteins and waste products. Among these, the proteins they contribute to least concentration, least of the osmolality of the plasma. The sodium it is the biggest contributor. Coming to the next question, the ECF volume of plasma. ECF volume it is determined by amount of sodium in the extracellular fluid. So ECF volume it is primarily determined by the amount of sodium in the extracellular fluid. So the extracellular fluid, we know that it's the fluid that exists outside of your cells in your body. These fluids would include the liquid part of your blood and the fluid that is surrounding your cells. So the question is, what determines the amount of ECF in your body? So plasma it is concentration of the plasma is not the primary factor that determines how much ECF you have. The amount of sodium you have in ECF, the more the sodium in your body, the more would be the ECF volume. So ECF volume is proportional to amount of sodium contained in the ECF and not to the concentration of these ions as per Guyton and Hall. Children and adults they have same concentration of sodium in plasma even through ECF volume are greatly different. The immunocytosis or reverse pinocytosis requires the calcium. So reverse pinocytosis, what do you mean by that? Let us understand this. Imagine a cell that has something inside a tiny bubble or vesicle and it wants to get out. So in a normal pinocytosis, the cell will bring things into this bubble, but in a reverse pinocytosis, they are pushing the things out. So the cell membrane, like the outer layer of the bubble, it fuses or connects with the vesicle covering. A small channel opens up between the inside of vesicle and outside of the cell. So the stuff inside the vesicle that wants to release passes out through this channel and goes out of the cell. So reverse pinocytosis, where the cell push material out of the vesicles, it involves the calcium. It involves the calcium ions. So inside cells there are vesicles containing substance that the cell want to release. To activate the release process, the cell need a signal. So here the calcium would act as a signal. So when the cell receives the signal, it allows the influx of calcium into the region where the vesicles are located. So this increase in the calcium is just like a green light for the release process. The potassium homeostasis in human is the potassium homeostasis in the human is mainly maintained by the fact that most of the potassium ions are found intracellularly inside the cells. So that is a key factor in regulating the potassium level in the body. While the other options like active secretion of potassium in distal tubule or maintenance of sodium potassium ATPase activity also play in potassium regulation, the distribution is the fundamental aspect of the potassium homeostasis. So which cells stop the leakage from the vessels? So these are the platelets. So platelets as shown in this figure. So when the blood vessels they are injured, the subepithelium is exposed. So the platelets, the cells that prevents blood from sticking out of an injured blood vessels. So they will form a platelet plug and hence formation of fibrin leading to the clot formation.